Thank you, Rohan. Thank you for the kind introduction, and thank you everyone for being here for talking. As Rohan said, yes, I moved relatively recently here to Montreal, so really happy to be able to talk again, from a different institution. All right, so I know it's a little late, so I'll try to to bring some enthusiasm to the talk today. And this is really something really basic that I found when I was working doing my PhD, and it has translated to the work I do now in my postdoc. And it's basically about how to do reproducible papers using R, but for the life sciences. So, probably most people here know about RMarkdown and know that it's really useful. We heard talked before today saying it's really useful to parameterize things, make things reproducible, and we're all aware of that. But the truth is that R is rarely used in the life sciences. When you look in biology or biomedical engineering or probably biology or stuff like that, people don't use R to create papers. They typically use other tools, and it's not the default method to create papers. So at the beginning, I was wondering, why does this happen? Why do people don't use R? Because once you use it, you realize how good it is, but most people don't use it. So going back and thinking about the things I was hearing when I was doing my PhD, some of the things were R is just for statistics, or the learning curve. It's easier to open some at that word processor and start typing than learning a little bit of code and how to change formatting with LaTeX. But I realized when I was using RMarkdown that one of the things I found really difficult back then, sure, I could create the documents, but the problem was, I need to create figures for a paper. How I'm going to create a figure if, I can use R to create just, outputs of data visualization or statistic summaries. But I didn't think it was possible to create, figures. So when I say figure, is because figures in papers in life sciences are usually, they can be quite large, but usually there are figures that combine images from cells or tissues, microscopy images, or images of mechanical load or something like that, or equipment. And the figure also has a data summary and something that someone did a statistical test, right, showing significance. That's basically how a figure looks. And how that look, a typical figure, I pulled this figure from a paper from last year, is basically something like this. You have here on the top panel, you have some statistic, right, some descriptors of your data. You have some Western blots or something going on here, and then you have images from cells or tissue and fluorescence images. So everything is combined and labeled in a figure, and this is what you want to produce for a paper. And when you look at this, you think, well, R is probably going to be good to do, the first part of panel B, but there's no way I'm going to be able to create all of this. So the question becomes, can you use R, and specifically RMarkdown, to create figures like this? And the truth is that we can use R to do it. And the way I like to think about it is that we can divide the workload in two parts. So one, is reproducibility, where we have, I particularly like Bookdown. I know people are moving to Quarto, but Bookdown has worked well for me, and it's great. So it's the tool I use. And we have package here, and I'm sorry for the developers of these packages here, PNG and Grid. I couldn't find hex stickers for them online, so I just made my own. If someone tells me where I can find them, I'll change them. But basically, the idea is we can use here and Bookdown to create a reproducible paper. And then we can use these four packages, namely Patchwork, ggplot, Grid, and PNG, to create a composite figure that gives you a result that is, publication ready. So we use the packages, and as I said before, Bookdown allows you to create a paper that you can break up, manuscript. You have an RMD file for materials and methods, for results, etc. And then if you want more details, because I won't have time today to go into details about it, there's a website, for Bookdown. You can find all the details there. And here, as probably everyone knows here in this room and there and virtually, here allows you to, not be struggling with, setting directories. You use here, and then you call everything, and you're set up. So let's think about a typical scenario. How would that look like for someone working in the life sciences? So you have written your paper sections. So you have your methods, results, discussion, and each one of these parts of your paper is a separate RMD file. And then you have some images, right? Some tissue images, microscopy images. but also you have some data that you're going to analyze, and you want to create this composite figure that you want to show in your paper. And then we want to create a composite figure. And you know, I have, at the end of the talk, I have the link for the repo where you know, everything is taken care of, you know, creating the manuscript and also creating the image. But today, for the sake of time, I will just focus on how to create the figure. So let's say you have a project that looks like this. So you have a folder with your project, and then you have a folder called data with your CSV files, because typically people will work with CSV files that they have collected. You have a folder or directory called code with your R scripts. You have one called figures, where you have your PNG files. It can be also, I have to mention, other type of images. What I've used so far is PNG. I know you can do the same thing with, you know, TIFFs or JPEGs. But that will change the code a little, but the take-home point is it's doable also. But for today, I'll use PNGs, and then you have a folder for your manuscript. So the cool thing about R is that you can read the image, and you can get the data, and you can do the analysis, and you can do all of these things in a single script, R script, and then you can call the script into your document. So we read the image using grid and PNG package that I spoke before. And then, of course, we use the true and known ggplot to create the plot for the analysis. And then Patchwork allows us to assemble everything. So how do you read a PNG image? So for example, I have this code here. So imagine that we have this folder called figure, right? That directory. Inside, we have a picture we want to put in a paper called cells, PNG. And then with here, we're gonna tell R, okay, there's inside this folder this file called cells PNG, read it, you know, with this function from the PNG package. And then this function here, raster graph, is part of the grid package. So raster graph takes the image and converts it into a graphical object, which the acronym, it's called GROB, I think. But the cool thing is that once you use this, you can actually call the object and make it interact with the other things you have done with ggplot. And then say you have your data, right? So you want to do what the example I have here, and I'll show you in a little bit, is we want to do a linear regression. So you have a linear regression, you have your data, and then you want to plot it. The thing most of the time is that it's easy to create the plot, you know, the linear regression, the line, the data, but how do you put your R2 values or your P values? So there's this package called ggpmisc. If you Google it, you'll find that the developer, which I'm sorry, I can't remember his name right now, but he created this interface, or created this function that allows you to extract from the model. He has a wide range of models that can be used for. You can extract, you know, the p-value, the R2, and it's going to be added to your ggplot object. It's really useful. And of course, the syntax will change a little bit if you're doing, you know, a linear regression, or if you're doing a t-test or something like that. But basically, you can extract the values, and this function allows you to put it onto the figure. And then the last thing we need, so we have read the data, we've analyzed it, we are reading the images. The last thing we need to construct our image is actually provide a layout. And this is explained in this, you know, this is actually the website for the Patchwork package. But basically, a layout is a way of telling how we want to assemble the figure. So this thing here is going to make sense in a little bit, but just keep in mind that whenever we see a letter, for example, A, we're just telling we're going to have we're going to have A, B, C, D, and E. We're going to have 5 things we want to put in our figure, and each one is going to occupy, you know, A is going to occupy the space, B is going to occupy the space, C is going to be here, D is going to be here, and E is going to be here. We're basically telling how we want to build the figure. All right, and then we want to use this function called wrap elements and with Patchwork, so we can actually bring the image, you know, to life. And this is just my cue to remember that I have to switch now screens, and I'm gonna go to. Are you able to see my the screen I have with code? I think you are, right?
Yeah. We've got your studio.
Okay, all right. So I am here now inside of the project, and I won't go into, you know, how you do everything, but basically we're following the same structure. You know, we have a folder for manuscript, we have a folder for images. So here's the script. So we're telling, and I think, let me see if I can make this a little bit bigger. Yeah, there we go. All right, these are to see for everyone. So we have some data, right? And we're telling our read the data we have, and we're just making some changes. We want to have a formula for regression. With A1, we're storing our ggplot. So we're basically taking our data and we are creating a linear regression, right? And we save it as an object or as a variable. With A2, we also, you know, to fancy and, you know, to do something else, we also want to do a histogram. So we have basically two outputs of data analysis or visualization that come from R itself. And then, as I was saying before, we have also figures. So I have, you know, this image we're going to call cells. We have another called molecule, and we have another other one called jellyfish. So we still are read them and make them objects that we can interact with ggplotting and patchwork. So we have 2 ggplot things and 3 images. That's 5 things we want to put into the figure, and we provide the layout, right? So we say one image is going to be here, which is with the first one, cells. So cells is going to be in this area. Molecule is going to be here. Jellyfish is going to be here, and the 2 outputs from ggplot are going to be D and E, and they're going to be here. And then finally, we just assemble the figure, right? So we call cells, then we call molecule, and then we call jellyfish, and then we call our 2 ggplot objects. And when we just provide some layout, right? Now, the thing is, right, everyone's wondering, how does this actually look, right? So I'm just gonna take a second. So I'm calling the document, everything is automated. Once you have the packages, it should work. Everything is running, the script is running, and we're gonna get an output, and the output is this. So we have a paper, everything is, you know, we didn't have to worry about margins, everything, RMarkdown took care of everything. And I didn't talk about this, but you can actually put, you know, other images if you want to put how your experiments were. But this is the main thing. This is how the final picture looks like, the final figure, sorry. It's labeled. We have our, I'm going to make it a little bit smaller. All right, so we have, you know, A, B, C, the 3 images. We have D and E, our output. We have combined everything. Patchwork provided the tags for the panels. We have our R values, we have our P values, we have, you know, everything is composed. We don't have to worry about margins or moving something and destroying everything when we do something. So it's automated and it works. So no matter how complicated your figures are, if you provide a layout, if you read the images and you do the analysis, you can do all of this. And we did all of this in a single R script. So I think this was for me the last hurdle I have to, you know, get over when I was working with RMarkdown. And with this, you are able to do everything up to the figure composition part within RMarkdown, and you don't have to be messing around with anything else. And it's automated, and you can customize it. You know, as I was saying, depending on the model you have, you can have different outputs here. But at the end of the day, the take-home point is it works. And it's once you learn how to do it and you get used to the, you know, the functions, it's really, really easy. So I'll just go back here to my presentation. So conclusion, you know, you can use R to create reproducible papers and complex figures. It's doable and it's relatively easy. And there is a learning curve. At the beginning, it's, you know, hard going from just doing, I wouldn't say the name, some text processor to a mix of coding and writing. But once you learn it and you realize the benefits, you never want to go back. And with that, I'd like to, you know, thank the people in my lab, Natural Lab at the Université de Montréal. And if anyone's interested, you know, taking a deeper look into the actual repo that has how to put up the paper and the figure and the slides as well, here is the link, and I'm going to put it also on the chat. And thank you everyone for attending the talk.